Thank you all for coming. It's great to see such a full room, and I'm actually feeling like a little nervous. I've given quite a few talks here now, but this is the last one. Yeah, it'll be about half an hour. I call my talk this because I think as academics, we get asked this question a lot. And if you're anything like me, usually I jump into some sort of technical description of my research at a level scaled for how boring I think I can get away with being. But it occurred to me that more infrequently, or much less commonly, do we get the chance to talk about everything that goes around the scientific side of academia. Like all PhDs, mine has been the journey of meeting people, discovering new research fields, traveling to new places. And if ever there was a day to talk about this, I feel like it's today. So just to confirm, this is not in any way a technical talk. I expect you won't learn any science from it, except for what I would say the stuff around the science that I think matters too. I agree with Will that we would not aim this at our examiners. I think I get enough time to talk to Tim about science after this, but instead we aim it at our parents. My parents that come to visit, and they much less frequently get to hear me talk. So that's the fun today, and I will try and show enough photos to keep us all from falling asleep. On that note, every photo on this slide here represents a person that I wouldn't have had the chance to meet, a person who I hold dear to me, had it not been for my choice to start in academia and do a PhD, or else it's someone who, frankly, I probably wouldn't have pushed through to the end of the PhD had it not been for that. So, yeah, for anyone wondering, that is Caswell lighting sambuca shots at the pub after his last student graduated. So it is always worth coming to the pub afterwards, just in case that happened. Okay, so I believe my academic journey started in 2018. I joined Andrew Thompson at Caltech, and his then postdoc, but now professor at UW, Georgi, in Pasadena, Los Angeles. And I arrived, I was sort of starry-eyed, I was really excited. It was my first chance living outside of the UK. And we were working on something to do with turbulent ocean fluxes. Andrew Thompson is an oceanographer, he's not a neuroscientist. I don't really remember much about this research, frankly. But at the end of the internship, what sort of has stood out to me and what I've reflected on is that by the end of this 3 months, I was pretty much ready to sign myself down, mostly out of momentum, to go and join Andrew for a PhD and sort of continue the line of work in oceanography that we've been working on. And Georgi pulled me aside one day, took me for dinner actually, and he said, in no unclear terms, that I really shouldn't do this. He said, if he had his time again, he would move into a field more like machine learning, that this was where all of the interesting stuff was happening, and that this would be a better decision for my career. It's really busy, so I don't know if people can maybe squeeze down on the sorry.
Oh, that's all. You don't know if they're upset.
Well, okay, I'll carry on. Not only was Georgi right, for me, but also just for the era that we're living in, what I've come to reflect on most is that it's actually very rare to receive advice like this. A lot of great mentors will tell you, you're doing great, carry on, you come and join my lab, or keep going with this project. But it takes a very special kind of person to look at you, know what's best for you, and tell you when it's time to leave. And so for this reason, I'm very grateful to Andrew and Georgi for this introduction to academia, even though I didn't end up working in their field. So I left Caltech. I did what any good scholar would do. That is, apply for a fellowship to work on a follow-up study, and then use the fellowship to do something totally different. In my case, it was to Harvard. Here I am with Sridhar and Will, two students. And I followed Georgi's advice and used this to switch into computational neuroscience and machine learning. To me, these seemed like complementary fields, as we all know. One is understanding machine intelligence, one is understanding biological intelligence. There's some synergy there. I wanted, yeah, neuroscience fit the bill for a field to apply machine learning into. It was relatively young, there was lots of new results coming out, and Harvard was and still is a leading center for it. So I spend most of my time sort of flitting between Genghis and Sam's lab and generally exploring the East Coast. So whilst I had a really amazing time at Harvard, I make no secret of the fact that actually I think I felt a little out of place there. Particularly maybe any Europeans who've had the chance to study or research on the East Coast will be aware that it's tough out there, and the culture around academia is different. It's not better, it's not worse, but it's different. And for this reason, I was really happy. I was really happy when I got an offer from the SWC to come back, return to my home country and study neuroscience in London's leading institute. It seemed like the best of both worlds. I moved into a flat with Will, who I'd known for probably 5 years at this point. He was studying at the Gatsby at the same time. And the first thing we did, as well as giving the Institute its first COVID scare, was Adam Camp's robotics course, which is going on right now. So here is Will training our first convolutional neural network on the robot on the left. And the result of it is on the right, which shows what I think we all knew, that neither of us are particularly human, but I am substantially more human. Okay, my next real job was to find some advisors, and I think it's fair to say I sort of took my sweet time over this. Claudia's smirking. I ended up with, in no insulting way, quite a hodgepodge mix of different advisors. This was more out of my own indecision making than any master plan, but I think 5 years down the line, I'm happy to report it was the best indecision making I made. So from the top, Claudia is a true computational neuroscientist who runs a tightly knit lab at Imperial. She is not only incredibly smart, but she is really caring and looks out for her students. She is also probably the most ethical and moral scientist I've had the pleasure of meeting, and I've been really delighted to have the chance to work in her lab. As for Caswell, how I met Caswell is a little unclear. He's not actually at the SWC. He sadly can't make the talk today. And he's an experimentalist, and I'm definitely not one of those. But when I was looking for a rotation advisor, people kept saying, you've got to give this guy a call, and you'll be a good fit for his lab. And I did. And like most people who intend to pass through Caswell's lab just for a little moment, I ended up staying there for the whole of my PhD. This welcoming attitude is, in my opinion, one of Caswell's greatest strengths and also flaws. He can't say no to anything, much to his own detriment, occasionally. Kim was based in London at the time, working at DeepMind, and she gave us a lecture, one of the introductory lectures we receive as students in the SWC. And it resonated with what few wisps of interest I knew I had. And I think it was Pierre, actually, I don't know if he remembers this at the time, who said, you're looking for an advisor, you should email Kim. And I'm really glad he did that. Kim is also one of the smartest scientists. She is wise. Is that enough for everybody? Very Kim. Yes. Is that a wall? I don't know if you want me.
Yeah,
press the
shut
power.
down. Yeah.
Power?
On.
It will shut down in 9 seconds.
Yeah, and then
She is wise beyond her years, and the choice to collaborate and work with Kim perfect, was, everyone good on Zoom? Okay. Was honestly one of the best ones I think I ever made. I could talk for half an hour to express my gratitude for these 3 people. That wouldn't make for a very fun talk, so I'll leave it with this and say that all of them have gone out of their way to back me up in a manner which I believe goes beyond the call of duty, and have majorly contributed to who I am and where I am today. So thank you. Okay, let's just reflect a little bit. I told you about how I found my research field. I told you about how I found my PhD program, how I ended up here. And I've told you about how I stumbled around and found some advisors. What I should do next is tell you a little bit about what I spent the bulk of my time doing. Before I do that, I'd like to just reflect and say, okay, there should be another big one. reconnecting to Zoom. Sorry about this. Sorry. Okay. I want to reflect on this and just say that when I present it like this, these first three steps can seem somewhat chronological and maybe even inevitable. And I think that might do disservice to the general stress, anxiety, and sort of perpetual sense of loss that, like many, I might say most PhD students or young academics feel as we're sort of moving into academia. And reflecting on this, I think it's partly because there's really no fixed recipe for ticking these first three checkboxes. And also, somewhat by definition, you don't always have good mentors, good advisors to help you through it. It's why I'm particularly grateful to the likes of Georgi, Claudia, Caswell, Peter Vincent, believe it or not, for taking the time to counsel me and advise me before they had even known me for very long. So my PhD can be broken down broadly into about 4 projects which I led, as well as a few side gigs that I probably won't talk about. These are all linked by the common theme of modeling the hippocampus. And again, this isn't a technical talk, so I will try and not get into too much detail. But I mean this in 2 ways. I mean, I build models. I spent a lot of my PhD building models to try and understand the brain, understand the hippocampus, comparing these models to neural data, and trying to understand what they teach us about the brain. And I've also spent quite a substantial amount of my PhD building tools which help us build models. So there's some good old-fashioned engineering in there too. And a very large, maybe uncountable number of people have contributed either directly to these projects or indirectly through discussions have affected my academic direction. And what I'll do now is I'm going to try and sort of move through these talks, through these projects one by one, to try and, I'm worried it's frozen now. Anyway, to try and assign some credit. Oh, it has. The slides aren't changing. It's a Wi-Fi. Yeah. Yeah, Zoom's disconnected. What I will tell you is that I'm going to zip through these projects, assign some credit to some people. I also wanted to point out that also don't be fooled. When you present work like this at the end of your PhD, it can look like there was some master plan to how these projects moved from one to the other. There was absolutely none of that in my case. A lot of these projects were created by happenstance, by single conversations, individual papers being sent. That's not something I'm proud or unproud of, but there wasn't a master plan, and I know it's that way for many people. I really apologize for these. Okay. All right, we're there now. Okay, so project 1, I had no idea what to do. And so I did what I think I'd advise all early PhD students to, and just ask. I asked Caswell for a project idea, and in turn, he put me in touch with Will DeCoffey, a postdoc in his lab who had a smart idea. Without getting technical, Will thought that the compressive effect due to a phenomenon called theta phase precession, prominent in the hippocampus, and a very simple learning rule called STDP, would combine to learn a type of representation called successor features. That's a lot of jargon that doesn't necessarily matter. What does matter is that Kim had relatively recently written a prominent paper on the successor features. And so it was in vogue at the time to try and understand how we could learn these things biologically plausibly. And over the course of 2 papers, we tested this, one paper more computational, one a bit more theoretical, and we discovered that Will's intuition was basically correct. And so I had to take home one lesson from this. It's that you don't know what to work on, just ask. Sometimes those low-hanging fruit projects, particularly early on in your PhD, can be a really good chance to get going and can end up being anything but that and can grow to substantial pieces of work. So that first project got me interested in oscillations and their role in learning. And in the second project, I was studying how these same oscillations can have another role in learning. Instead of exploiting the compressive effect of these theta sequences, I was looking at how these oscillations can coordinate information flow through neural networks in the brain in order to guide learning in a much more general sense. That's pretty much all I'll say on the technical side of this project. But what I will say is that around this time, I happened to be in Okinawa. I was working with Tomoki Fukai, arguably my fourth advisor, and I had originally intended to work with him as an internship after Harvard, before I moved to UCL. But due to COVID delays, I didn't get to travel there until middle of 2022, and I'm happy that the SWC basically let me go and do some of my work in Japan for no particular reason. Before going there, I didn't really have a clue what to work on. And a shout out here, actually, it does have to go to Peter Vincent. He will probably remember this. One day he sent me a paper, a random link. He said, I haven't really read this, but it kind of looks up your street. And I did read it. And yeah, it played with a few ideas that I've been thinking of. And then by the end of the afternoon, a clearer idea for this project was in place. I only say that because it's a bit cliche, but sometimes these single interactions, those individual papers can lead to something much more. And I think these happen more frequently at institutes focused on collaboration like the SWC. So I was giving a lab meeting in Tomoki's lab, and to zoom back a bit, for these first two projects, one thing I'd been required to do was make a simulation, quite a trivial simulation, of a little agent moving around a box and the activity of some place cells and maybe some grid cells as it did that. And at the end of this lab meeting, two very close friends who were in the lab, Gaston and Tom, came up to me and they said, project looks cool and everything, but we really like those little simulations you made. We work in the same field, and we're also doing a lot of this kind of simulations. It would save us a lot of faff. You could tidy it all up and publish it for other people to use. I'm grateful they did, because within 3 weeks, the rat in the box package, as we called it, was live. And what started as just a little bit of code on my computer to simulate some trajectories in 2D grew through my own, but also through a lot of community contributions into quite a substantial platform for modeling many aspects of spatial cognition. We now have users all over the globe and have something like 60,000 downloads. I'm not sure why. I think because it has a silly name. This is the piece of work that has somewhat come to define me, I think. Not in my head, but I think for other people. I see Peter grinning at the back. What I will say on this is that in comparison to publishing sort of more traditional papers where you're releasing a set of ideas or a set of results, making tools, particularly tools that people use, has sort of catalyzed in my experience, very rich interactions and engagement with the community that I found very satisfying. On this front, I do really want to thank Colleen and Quinn. Quinn is starting his own lab now over in Canada, who I think that there are many people here to thank, but who were early champions of the package, integrating it into their own projects, and more importantly, encouraging me to turn this into a big sort of open source community endeavor. Colleen is a big proponent for all things open source. So thank you for this guidance and advice there. Okay, if you didn't see, project 2 and 3 were done in about 4 months during my time in Okinawa. I don't know what I learned from this other than productivity really ebbs and flows. Many people will have experienced this. And if you want it to flow, a remote tropical island where there isn't much to do can be a good choice. Okay, this last project was by far, I think, my favorite. Mammals rarely ever think of where we are right now. Instead, we spend a lot of time thinking of planning a future route or remembering where we were within these silly things like theta sequences. And I had become concerned with how this non-locality was affecting the measurements of John O'Keefe's famous place cells. To put it simply, it blurs them and distorts them. And it's likely that this phenomenon I spent a lot of my PhD studying was actually blurring the place cells we're trying to study in the first place. So in my head, I think I'd sort of come up with an algorithm that would solve this. It was easy to implement and had some empirically good results, but I couldn't prove that it would work. And that's a problem. I was just going off a hunch. I was making coffee one day in the kitchenette area, Pierre walked through, and I got talking to him about this project. And I think by the end of the coffee, he was so frustrated with the shoddy attempts I was making to prove this that he agreed to come on board the project and do some heavy lifting on the maths front. I think that's fair to say. Will attest to the fact that working with Pierre is one of the most intense processes you can go through. It's also, I say without a shadow of a doubt, the single most fun paper I've ever written. And it was a total joy to have the opportunity to work so closely with Pierre. So if you can't prove it, I do recommend just trying a Jasper student every now and then. Okay, so that leaves us pretty much done. I'm definitely almost over time, but I just want to say a couple more things. Before I finish, I don't think I can leave without talking about the Camina. So 3 years ago, thanks to Kim, actually, I came into contact with these 5 amazing scientists from all across the world. And we set up what is now called the Camina. It's a summer school hosted in Africa for students across Africa to teach the basics of computational neuroscience and machine learning. Despite having a surplus of very young and motivated students all across the continent, Africa simply, for many reasons, doesn't have, computational neuroscience doesn't exist in Africa to the same extent it does in the West. We're already in our 4th year now. We've raised something like $150,000, and we've hosted courses in Ghana, in Rwanda, and in Zambia to meet and teach students who otherwise may not have had the chance to access this knowledge, and more importantly, to join our community. It's very possible and only possible due to our sponsors, and I do want to say thank you to the SWC, who have given consistently and generously to this course and asked for very little in return. It makes me proud to know that endeavors like this to make science more fair and more equitable are still supported, not just in my country, but in my own institute, particularly with the global climate as it is. Just to point out how one of our students, Vincent, and how the course can kickstart careers. This is Vincent. He participated in the Rwanda school, and before this, he'd actually never left his home country of Uganda, where computational neuroscience doesn't exist in the same way. Since the course, partly thanks to a separate scheme run by Kim, he won funding to attend COSIGN, which is the big international conference in our field. Here he is with Kim and I and Leah. And since then, he's also been accepted onto another big prestigious international summer school called the Imbizo. He's applying for international internships at OIST, of all places, and prestigious international PhDs, and I'm very confident he's going to get accepted. People like Vincent remind me that sometimes it's that initial opportunity, the summer school, in my case, a little internship at Caltech, that can kickstart careers in academia. Funny story. I got to the conference, I give Vincent a big hug, and I said, "How's the conference going?" And he said, "It's good, there's only one issue. I can't go outside I can't go out and join people for dinner because I didn't bring a coat, and it's really cold." I said, "Of course, it's Montreal, it's the end of winter, what did you expect?" It seems obvious, but Vincent had never left Uganda before this. I just simply didn't own a coat that would be warm enough for Canadian winters. I tell this story with Vincent's permission because to me, it's a good reminder that sometimes the barriers to science aren't as obvious as just financial or access to institutions. We gave him a coat, we found him quite smart, but we found a big Canada Goose jacket, and he walked around the conference wearing it all day. He looked great. So he did get a coat. All right. Final slide. I just want to say a few more thanks. Firstly to the SWC itself, which not only encourages international and external collaboration, but with the likes of April, Clara, how would I not mention Karen, makes doing so really, really easy. I also want to thank a few people who don't usually get thanked in the context of an academic talk like this. First of all, I think it's my family. In particular, I'd like to thank my sister, who made living in London for 5 years a real joy. She was a big part of my support network. She's not here today because she finally quit her unsatisfying corporate job and started a postgraduate medicine degree at Manchester yesterday. It was a tough decision, and I'm very proud of her for doing this. It means that I won't be the only doctor in the family for very long, but I will be milking them. Lily and I met long before I knew what academia was, and before she probably knew what she was getting herself into. Thank you for being by my side, for rolling with all of the uncertainty, and for never minding that I don't really know what the hell I'm doing in 12 months' time. I don't expect that to change, but it's been wonderful having you around. But a fun fact about Lily, as a side gig, so working as a prison officer in the UK's biggest male prison, full-time, she researched, wrote, peer-reviewed, and published a paper on the use of language in male prisons. This paper now has more citations than a vast majority of my own, and that's a testament to her intelligence. As well as that, she also survived, maybe I could say enjoyed, flat sharing with Will and I for the best part of half a decade. And I think for these reasons, she probably is deserving of her own PhD. Finally, after a lot of reflection, the most important person I need to thank is actually Will. There's a running joke that whatever Will does in his career, Harvard, Okinawa, UCL, I follow him shortly after. So I'm grateful for him letting me give this talk first. And the funniest thing about this joke is that it's true. And who can blame me, because anyone who knows Will will be aware that he is possibly, quite possibly the smartest scientist of our generation. And I have known Will for a long time now. We've studied, traveled, lived together for the best part of, for over a decade actually. Thank you for being a great friend, but more than that, also a great role model. I will be genuinely sad not to live with you next year, but I look forward to following your career and finding out around. With that, I'll say I'm off to do a postdoc in Montreal. Lily's also moving to Montreal, we're going together. Come and visit us, and at the least, come to the Exmouth Arms tonight at 6 p.m. or up here around 4:30, where there'll probably also be some champagne if everything goes correctly. Thank you. I guess I should say I'm happy to take questions. There wasn't much science to do a question in, but if there are any, meta scientific questions, or feel free to ask. If not, you can ask me. Do you get going? Yeah. Thanks, everybody. Good.
Can someone on Zoom and say if they can hear me? Great, thank you. Okay. Matching Tom's description as quite significantly less human, I'm going to do a science talk. It's aimed, yeah, you'll see it's not my normal science talk. Okay, let's go. So, for people who don't come to science talks normally, don't just sit there. Please ask questions and heckle and stuff. Some things that scientists like to do with their questions are ask confusions, or ask a question that's not really a question, but in fact a comment on how they broadly disagree with the person's entire thesis, or just trying to appear clever. So if you want to try any of those tactics, you're welcome to. Okay. So in this building and in my PhD, studying neuroscience, so it's the study of nervous systems. You all have a nervous system. This is your brain, your spinal cord, your nerves, and they do. What do they do? What are they for? I think this is best illustrated by the animal you all know and love, the sea squirt. So on the left is a juvenile sea squirt. This animal has a nervous system. It looks like a little tiny tadpole thing, and it uses this nervous system to control some muscles and swim around, and to sense gravity and light. And then it uses those impressive nervous system to find a rock, attach to it with its mouth, and then never move again. And it turns into this like plant-looking thing that filters the water to feed. And in doing this transition, it recycles its nervous system. Once it's attached to the rock, it no longer needs it, so it eats it. And so this illustrates really nicely what nervous systems are for. They're for controlling mobile multicellular organisms. And when you don't need one, biology, evolution destroys it. And with this, they do some incredibly impressive things. I think there's no end to the number of things you can point to that are impressive, but every one of your sensations and perceptions is the result of your nervous system. Across biology, you find crazy examples that go beyond what we're able to do, like elephants who can sense low rumbles across the ground through their feet, to moths that can sense up to like 300 kilohertz in order to drop and avoid a bat as it comes flying past with its echolocation. Spiders have a much better sense of touch than us, and they use it and extend it over their net in order to sense what's arriving onto the net. We use it to do impressive motor actions, so the animals in trees or hovering to eat from plants. And all of your thoughts, your sense of yourself, your ability to play chess, your ability to join the orca whale pod and hunt seals using culturally specific hunting patterns, all come down to our nervous system. So it does some very impressive things, and it does it in some very puzzling ways. So I think if you ever lack wonder in your nervous system, you can go read the books by Oliver Sacks. One of his most famous is called The Man Who Mistook His Wife for a Hat. So this man had some damage to his fusiform face area, which is an area of your brain that processes faces. And that meant that he was, despite the fact he was able to see, he was unable to do the process of sticking together the parts of his vision that form a face to recognize faces in others. And so he was able to do what to us seems like completely outrageous. He was able to look at the scene and see the world, but unable to stick things together to make the face of his wife, and able to interpret instead a hat as being the best approximation to his wife in the room. And so really, I think it's hard not to be presented with all of those facts and not wonder how this thing is working. And if you're slightly more, mercenary, there are also some good uses you can get from this knowledge. So not only for curiosity and asking some like really deep philosophical questions that people have pondered over through all of existence, but you can do things like create medical technologies like cochlear implants that allow people to hear, or speech prosthetics that allow people to speak. And the very first like artificial neural networks were all based on our early understanding of the nerves. So hopefully you're well motivated that this is a reasonable thing to spend 5 years studying. So my question is, how does this work? I'm now going to give you one level of a basic answer as a way to give you some background material that will be required through the rest of the talk. So pay attention. Here's basic answer number one for how the nervous system produces everything that makes you you. I'm going to give you two things that we're going to use. The first is that the brain is really surprisingly modular. So there's a bit on the top that is called motor cortex and is involved in coordinating your limbs. There's a bit at the back called V1 that is involved in processing visual inputs, a bit that does auditory, a bit that does language. And so you can postulate the function of the brain into these different brain areas. And it seems like there are meaningful little computations going on in small sets, in small areas of the brain. And the second thing you need to know is that the entire nervous system is kind of defined by being made up of these cells called neurons. You have something like 100 billion neurons in your nervous system. They're little cells that have inputs. They integrate all of these inputs, which are patterns of electrical activity. They process them somehow, and they have a cable that heads out the other side and sends some pattern of electrical activity out. And the key thing about these is the language they speak here, as I'm describing, is all these patterns of electrical activity, which are these like pulsatile clicks of spikes that a neuron will send. So the old school recordings, if they like stuck an electrode in, they would hear that like click of the neuron pulse. And so the key thing is that like there are all of these 100 billion neurons, but they all speak the same language. So if you look at the neuron that connects your spine to your toe, which is the longest neuron you have in your brain, or the one that connects your eye to the visual cortex, or the one in your prefrontal cortex, they're all speaking the same electrical language, but using it to encode different things, like curl your toe, there's a particular visual feature over here, or remembering his name is Tom, an important thing for the current situation. So that's level one of understanding what the nervous system is doing. Now I'm going to try and get slightly more precise to describe what we've been doing in my PhD. So broadly, we think the brain is computing and is doing it with these small little chunks that are doing meaningful algorithms, and it's using neural activity, this pattern of electrical activity, the language of the brain, to do this algorithm. And what we'd like is some kind of link, something that tells us how the computations that are going on inside the brain are related to the neural activity we measure. Currently, that seems like an insurmountable gap. How does our consciousness arise from patterns of electrical activity in our brain? I won't answer that, but we might get to a smaller version. So there's loads and loads of streams of evidence. You've heard about like what Tom did in his PhD, which is part of this field. And I've also talked to you about lesions, where the fact that removing that brain area, remove facial perception, gives you a clear hint about what's going on in that brain region. Here's another one that I put in just, I think, very cool. Comparative evolution, you can look across two closely related species. For example, here's a trout and here's a murmurid fish. I believe they're cousins or something. And then you can look and see huge differences in their brain architecture. So this one has an enormously expanded cerebellum. This is whole structure on top that is as big as the rest of the brain put together. And so for some reason, evolution has decided to stick an enormous cerebellum on the back of this animal. And the big difference between these two fish is the water in which they live. Trouts live in clear streams, while murmurids live in turbid waters where you can't see very well. And so to evolve for their niche, they've built an electrosensing location that they can use to detect the electric fields of fish around them that they can use to then catch and eat them. And it turns out that the way nervous systems process such electric sensing is with a cerebellum. And so you can go and say like, oh, that cerebellum must be there, because in every species where there's this type of electrosensing, we see this structure that performs this kind of electrical computation. And it's not present in other fish that don't have that. So, hopefully that's illustrative of a couple of ways you might try and build that link. I don't do those either. So, what do? I try and build, so I'm going to Gatsby, so I try and build mathematical theories. And these mathematical theories aim to be predictive, interpretable, and generalizing. So I'm going to try and illustrate an example here of what we mean. Perhaps the gold standard for building a mathematical theory in science is Newton's laws. This describes the motions of objects, and it does it in a very predictive way. But it also has these features that it's very interpretable. Like when I first learned about Newton's laws, I didn't really get the concept of a force. A force is to me, quite an abstract concept that changes the way you look at the world and gives you tools for thinking about it. And so it's very powerful. It gives you these new ways of looking and seeing the world that enable you to understand and predict what's going on. And it's incredibly generalizing. Like Newton was able to use the same ideas to explain terrestrial things like balls rolling down slopes and celestial things like planets going around the sun. And I think it's very profound that philosophically profound, that the same ideas can generalize to both those contexts. And so we want something like that, but in neuroscience. And so the way, I think the best candidate we have are normative theories. So the norm in normative is like a moral norm. It's a thing saying this is what is good and it's what you should do. And so a quick example of this, again, that I'm not going to use, is about wiring. So your brain has all of these wires that are connecting things together. And broadly, all things being equal, you should use as little wire as possible. Because it's costly. If you build more of it in, you have to spend more spikes, which cost more energy, and it makes your brain bigger, which makes birth harder. It's all just bad. And so you should use the option. For example, why is the fusiform face area, the one that processes faces, roughly where it is in cortex? Well, what do you need if you're going to build a face area? You need access to visual information that's being processed a lot, so you can extract the face, and you need access to the memory system, so that you can remember the links between that face and the rest of your life. And so you maybe want to put it roughly at the end of the visual processing pathway and near the memory system. And that's roughly where you find the fusiform face area. And this is a bit hand-wavy, but versions of this theory get very precise and can predict exactly where C. elegans' 392 neurons should be in its body in order to minimize wiring. So a normative theory is one that takes this goal, like minimize wiring, and uses it to predict the behavior of the nervous system. So we would like a theory a bit like this, but that does the mapping that I've been talking about throughout. We'd like something that goes from algorithm, from the computation we think this brain area is doing, to the neural activity that we measure. And the basic underlying assumption is that if we knew the algorithm this circuit was doing precisely enough, and we had some idea about how brains tend to implement algorithms, we should be able to predict what the neurons are doing. That's the assumption. So that was my long-winded introduction. The only thing you need to remember from all of it is that we're doing this game, and hopefully you're motivated about why it's a reasonable game to do. And then what I'm going to do for the remainder of this talk is I'm going to describe the particular algorithm and neural data I spent a lot at the beginning of my PhD thinking about, two models people have in the past used to understand this, what we did instead, and then some output. So let's go. This bit is the bit that most requires you to remember things. So this is another pay attention section. It will be used 3 times over, so it's really worth the investment. Okay, so we're interested in linking algorithm to neural activity. So I need to make precise what I mean by both of those two things to turn this into something useful. So on the left, I'm going to describe the algorithm I mean, and the algorithm I am going to talk about is path integration. By this, I mean your ability to wander around and understand the structure of 2D space. For example, if you start, maybe you've never visited this institute before, but you start In the institute, you head one step west, one step north, and one step east, and end up over here and realize you need to come back straight away because the drinks are about to start. Then you don't have to take the path, you don't have to just remember where you came from and retrace your steps. You understand the structure of 2D space and are able to take the shortcut that will take you straight back home. And the reason you can do that is because you have the understanding of the structure that you're at from your current position. If you were to step one step south, you will end up back at your home, back at the institute here. And so it's the ability to make these hypotheticals. From my current position, if I were to take this step, I would end up there. So that is the algorithm we want this circuit to perform. And there's a lot of very good evidence that I won't go into that this particular brain area I'm talking about, the entorhinal cortex, does perform this algorithm. And there's a lot of very, very beautiful neural data about how this, what this neural activity looks like in the entorhinal cortex. So we're going to see a few of these plots. What I'm showing you is this is a square room viewed from the top. The gray lines are the path that a rat took as it ran around this room over a recording session, something like 40 minutes or an hour. And what the experimenters did, they put an electrode in the entorhinal cortex, and in this case, they're measuring the activity of a single neuron, and they're putting down a black dot every time that single neuron spikes. And so what you can see from this plot is that one neuron activates whenever the rat is in a triangular lattice of positions. So this neuron fires when the rat is in all of these positions, but anywhere else in the environment, it doesn't fire. So hopefully you'll agree this is like very beautiful. It's also very puzzling. Why on earth would a neuron deep inside the brain choose to encode the rat's position using this very, very puzzling form? Why is it encoding space in this periodic manner? There's two more layers of structure that we're going to talk about these grid cells, because there's not just one grid cell. Turns out they come in groups made up of hundreds of grid cells. And so here, for example, are two grid cells from the same group. We're calling a group of grid cells is called a module. So these are all from the same module. And you can see that these two neurons, they look basically the same, but all you have to do to get from one to the other is shift the pattern, translate it around, move it around, while keeping the rotation the same. So you can see, I've drawn it on here, the underlying lattice is like this structure is the same between the two, but it's just made by like shifting it around. And so what you find when you go looking is this is a schematization of one grid cell that activates in all these positions. And there are hundreds of them in the same module that have the same receptive field, just translated a little bit. So this blue one has the same one, but shifted down and right. And the green one has the same one, but shifted up and right. And overall, you find each of these neurons will tile a different bit, and together they tile the whole of space. Okay. This is a key bit of the structure. They're same lattice, translated, not rotated. And then finally, there's not just one of these modules, there's a handful, something like 5 or 7 in rats, and they're defined by having a different lattice. So for example, here are 4 from 4 different lattices, and here's a schematization of 3 more. So overall, to summarize, the structure is there are something like order 1,000 grid cells maybe in an animal, and they're broken down into a handful, roughly 7, maybe modules, each of which contains hundreds of neurons that have just a translated version of this hexagonal. That's good. And then our question is clearly why? Well, it's very weird, and it doesn't make any sense. So we'd like to understand why the brain has chosen to do that. So to clarify, we're looking for a normative theory that gets us from the idea of path integration to these grid cells that fits our criteria we're outlining. Okay, good. So now we're going to talk about how 2 existing theories try and tackle this problem, and then what we did in response to it. So the first theory is called the efficient coding hypothesis. This is a really old and successful theory. It's been around since the 50s, basically as long as people have been recording some awake behaving animals. They were instantly like, this is a reasonable way to think about neural firing. And the way to understand it is by thinking about sending telegrams through an undersea cable. So to do that, you need to find a way to turn the messages, i.e. each letter, into a pattern of electrical activity that you're going to send through the cable. So for example, H might become the binary 101, E to 01. And any pairing that takes a letter and gives it a unique binary code will work. You'll be able to send messages. But space on this cable is really limited. And so you want to design your encoding of letters in such a way that you can maximally pack information through the cable. And so it doesn't take too much thought to conclude that a letter like E that occurs most often in English should be given a really short code, one of the few short codes you have. and a letter like Z that occurs very rarely should be given a long code. And that way, by building an encoding like this, you'll be able to most efficiently transmit information through the cable. And you can hopefully see the analogy that goes on is if you replace the sender with the eye and the receiver with the brain, then what's going on is the optic nerve that connects your retina to your visual cortex is sending images through the cable, and it's transformed. It does it in neural activity. That's the signaling mechanism. And we should find that the neural activity is forming an efficient coding of these images in the same way that this binary pattern can be understood as forming an efficient coding of the letters. And yeah, I think this is a really, really great theory. It's very beautiful, it's very predictive, it's very interpretable, and it generalizes a lot. Like we've already shown how the same idea is generalized between an undersea cable and optic nerve, so you can believe that it also generalizes to other parts of the nervous system. Pretty nice. Unfortunately, it doesn't really explain the recordings I've been showing to you, about the fact that people have tried to use ideas like these for the last 15 years to understand grid cells. So if you take, doesn't look great on that screen, but if you take a code for space and try and optimize to encode position really, really well, you don't get grid cells. You get these random multi-field cells, which means that they activate in multiple positions, but those positions are not arranged into a grid. And on reflection, this should make a lot of sense. So I hope I'm going to explain, if it's not already obvious why now, is the problem with having a neuron that activates in many positions is that if this one neuron activates, you don't know where you are. You know you're in one of the places in which this neuron activates, but you don't know which one. So you could be here, you could be here, you could be here, you could be here. So what you need is a second neuron that can distinguish those hypotheses. So for example, in this case, you have a neuron that activates here, 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 but the only one of those that overlaps with its neighbor is that one. And so if both this one and that one are activating, you know you must be in this position in which both of them activate, rather than all of the other places where each of them activate alone, but without the other. So ideally, if you're going to build a really clever code of position, you would build it like this. But instead, what the nervous system does is it constructs many, many other neurons, all the members of the same module, that don't distinguish what their friends are doing at all. If this orange neuron is firing, you know you're in one of these fields, but but the green neuron cannot distinguish any of those positions. It's always off for all of the positions in which the orange neuron fires, because it's just a translation of the same pattern of activity. And so rather than creating the optimal neurons for encoding space, the nervous system has created hundreds of copies that are a waste from an encoding perspective. They confer no extra coding efficiency on the representation. Yeah, and so this theory does not work as a theory of grid cells. And the reason it doesn't work is because it doesn't capture this aspect that we've been talking about throughout, is that we should have some idea of the computation that's going on. The algorithm that's being performed in this brain area should be relevant for predicting the firing of these neurons. And we haven't even mentioned path integration yet. And now I'm going to try and persuade you that the most natural way to think about these grid cells is because they allow you to do exactly the computation that I've been outlining. So it is true that if this orange neuron, perhaps you're actually here in this grid field, but if this orange neuron fires, you know that you're in one of these places, but you don't know which one. But what you do know is that if you step south, the next neuron that should fire is the one that has the same receptive fields, but translated one step south. Okay, so the translated structure makes it it's not great for encoding, but it makes it trivial to do path integration internally. Because for every step that you could possibly make in the world, there'll be another grid cell with the same firing patterns translated by the same amount. And you know that that's the neuron that should activate, regardless of which one of the places you started at. It will always be the same. Okay, that's the whole point.
Well,
yeah.
So random fields really have higher information?
Yeah. But this is like this is me numerically optimizing. That's my advisor who knows my work very well. Numerically optimizing the code to be efficient leads to cells that look like this. Yeah.
That's a numerical result.
Yes.
Do they have to fire you?
Okay. And then you get left with one final puzzle, which is, sure, you can do this path integration thing, but I still have not answered the question of, you still need to know which of these ones you started in. And together with the way that this gets solved by the grid cell system is that there are these other modules. And so you can use those, like I was talking about the use of these pair of neurons up here. Together, they can help you distinguish, like if this neuron is firing and that one, you're probably in this field. It's the only one that overlaps. So it solves both the coding question while doing path integration. Okay, good. So what we need from this discussion is theories that include computation in the idea. So I'm going to quickly go through one, and then our answer, and then wrap up. So that was the efficient coding hypothesis, which is the best studied and most established of these theories. The alternative that's existed since about the 80s, I guess, is connectionism. This works as follows. You take whichever algorithm you're thinking about, you train an artificial neural network to perform that algorithm, and then you look inside the network at the neurons, and you see if they look like the ones you have in the brain. And if they do, job done. That's your model of the brain. And these neural networks can do basically everything, like ChatGPT is one of these neural networks. And so they're very good at modeling all of the different tasks you might think a brain is able to do. And this process kind of works. So just like not long before I started my PhD, people showed that you could train these networks, and they did indeed. These are like the same plots showing the activity of the neurons across space, and red is highly active and blue is not active. And you can see that they have these lattice-like tuning curves from one paper from DeepMind and another paper from New York. So that's big success and gives us a much simpler model to study. But unfortunately, what we want is theories that tell us how the system works in ways we can understand. But we understand artificial networks about as well as we understand the brain. We don't really. And so we don't know why ChatGPT does what it does. And this is bad from a scientific point of view, where our goal is to like understand the world. But in this case, it's actually bad not just from an aesthetic perspective, but it turned out that this one is not actually a good model of grid cells. It turns out if you just get noise and filter it, you get a representation that is as grid celly as the ones they reported in this paper. And so this is from the world's preeminent AI company in 2018, and they're not able to distinguish noise in their model from an actual grid cell representation. So I think this is like worrying for your theories. And these do better actually, but they're still square and not hexagons, and it's really unclear why that's true. So what we want is theories that don't just match the data, which turns out maybe they don't, but in ways that we can understand so that we can look out for these errors that are just really easy to make with models that are really, really complex. Okay. So that's what we tried to do. We tried to build a theory that was as interpretable as the efficient coding hypothesis, but with computing ideas just like neural networks have. And so the way this works is we try and find an optimal encoding that faithfully encodes positions efficiently. So this is just the efficient coding hypothesis. It's like your undersea cable encoding position, but it does so in a way that lets this computation happen. And so what does that mean? It means that whatever your neural encoding of your current position is, you should be able to predict from just some neural encoding of which action you're going to take where you end up. So this is the math equation, but I don't need to go into it. But if you, this is me numerically optimizing a code with this added constraint that you can do path integration. And what you find is you get multiple modules of axis-aligned grid cells, exactly the 3 layers of structure that I was talking about at the beginning of the talk. These are 15 neurons and how they activate across a square room. The green means highly active, the blue means not active. As you can see, each of these neurons has a lattice structure. They fall into groups that are translated versions of one another. And some of the lattices are quite hexagonal, not all of them. So I guess it's not a great theory in that respect. So it kind of worked. So hopefully now we're closing out. So as a theory, it does the job of being interpretable. I haven't really shown you that's true, but it turns out if you give me much more time, you can talk through every step of why each piece of the structure arises from the maths we've written down. It does it in a predictive way, so I'm not going to have time to talk through this, but you can predict things like the angle between successive modules should be roughly 4 degrees. Who knew that? That's great, and turns out it's roughly true, so that's even better. And there's lots of other predictions that we've been trying to test. So here's a mouse on top of a pyramid for non-obvious reasons given the talk, but it's real science. Okay, so that was, so it fits these first 2 criteria, but it also does it in a way that generalizes. So I spent the whole talk talking about entorhinal cortex and grid cells, but actually I spent most of the last 2 or 3 years thinking about other things, in particular frontal cortex, and how in frontal cortex structured working memory algorithms might operate. And this, things like remembering a phone number, in which you have to remember both the numbers and the order in which they came. And it turns out there's some very particular neural structure in frontal cortex while these algorithms are being performed, a bit like the very specific structure in grid cells while path integration is being performed. And super similar ideas to those we were developing for the grid cell theory can be used to try and understand this map. So these ideas generalize across different brain regions into different algorithms. So yeah, the goal is some kind of framework for predicting neural activity from the computation that's being implemented. There's loads and loads of stuff to do, like work on the theory and work on what the algorithms are, so the whole thing. But hopefully it's persuasive that normative theory can help you in understanding what the brain region's doing, and in fun and non-trivial ways. Like if you have a hypothesis about what the brain region is doing, you can build a normative theory, and if it doesn't fit, it means there must be other stuff going on. And that is in fact exactly what happened in the frontal cortex. There's whole layers of structure that we have not been able to understand that prompt us to think more about what the algorithm that's happening inside that brain region is. And I think it's kind of magical that you can scratch symbols on a piece of paper and somehow make predictions about, that was great, and I'm very grateful for the generations of people whose shoulders we all stand on making that possible. So that's me done. Let me just thank some people. Oh, no, I have one more thing. So a lot of my hairs have been going white during the PhD. I thought I'd show you one that Nish and I found yesterday that was particularly funny. I don't know if that's the picture, it's not very clear, but it's got this weird variegated pattern where it goes black then white then black then white. And I think it's a good analogy for the procedure of the PhD, which started off maybe okay, and then it was, oh, what's going on? And it kept that for a while, and then I had to write a thesis, and that was really not fun. So that's that thing. So yeah, it's been stressful, but I'd like to quickly thank a lot of people. I'd like to thank my 3 advisors, Peter, Tim, and James, for all here. I think I've been very grateful for the mixture of freedom and support that I've been able to have. I've been both able to disappear for months and do whatever I want for a while, and then come back and be schooled on how terrible at linear regression I am by Tim quite regularly. And that combination has been excellent. I'd really like to thank all the people I worked with. There's been a lot of great collaborations with the PPC team or the grid cell team, who I'm especially grateful for the time when I was lying in bed, completely unable to think with brain fog from COVID, and they just completely took over the whole thing. That was amazing. There was an undergrad who worked with me, which was very brave, so I'm grateful for that. And the modularity team, in particular, Kyle, who I worked with in Stanford and then on and off since, who's been an amazing collaborator. And broadly, the community around the Gatsby and the Barron's Lab for providing a super stimulating place to work. I'm also, very grateful to all of the friends and family who've made the last 5 years a really fun time. Yeah, I think there's a lot of people in there. But I've really enjoyed the last 5 years of my life, and I'm very grateful to all the people who made that possible, especially my family who supported me not just the last 5 years, but the last 28, including helping me dig ponds to put tadpoles in when I was 10, hosting chemistry-themed birthday parties when I was 18, turning up to watch me prattle about neuroscience when I was 28. So all of this. So thank you very much. Done. Yes.
So with your, I can't remember what you called it, the pathway recognition, I can't remember exactly what you said. Your single neurons firing, and it's creating this lattice pattern that you deem as inefficient. What? I know it's not really the purpose of it, but what's your best guess as to why that happens as opposed to a more efficient system?
Unfortunately, I had to remove all of that stuff from the talk, but it takes a while. But so it's true, it's not the most efficient, but it turns out it's quite efficient. So it's like a good answer, especially when you combine it with this idea of being able to do path integration. So it's like this picture is like, sure, it's not the most efficient way to do it, but the one thing it does let you do that other codes are not good for is working out, if you take a step, where will you end up? And so and that's what the theory is trying to show, is like, if you add this extra constraint that if you take a step, tell me where I'm going to get to, then given that constraint, this is the best code you can have. So it's the combination of trying to encode space, but subject to the constraint that you can do this very important computation that we really think is like the key thing that's going on in this circuit. Yeah.
I see that it's nice that you have these like codes that repeat, but why do they have to be hexagonal? Like you could have a place cell code as long as there's another place cell.
They don't. Yeah, and so it depends on how you set up the problem. The thing that in the theory produces hexagonality is quite contrived, but it ends up being a mixture of you get a low frequency bias from the size of the room. Like if your code was very, very high very, very low frequency, very, very high frequency, you need the two length scales in the problem. There's the length scale of the room and the length scale that you care about distinguishing. Like you're going to chop up space into some resolution. And this one pushes you to have high frequencies, those that oscillate quickly enough that they distinguish points inside the room. And this one pushes you to have low frequencies that oscillate slowly enough that they don't worry about encoding the points within one resolution differently. Yeah. And so you get a low frequency bias from one direction, a high frequency bias from the other direction. That gives you this like golden ring of frequencies that you want, which are in the middle. And it turns out the best way to pack frequencies into that annulus is to use the dented packing lattice in 2D space, which is a hexagonal lattice to get grid cells. That's like a super speedy version of that story. But yeah, there is a logic to it, but it's completely separate to everything I've talked about.
Yeah.
It's not a book. Yeah.
In your motivation for, minimizing wiring like the free spawn phase area, it was like a kind of a network embedded in 3D space. All your neurons, in some sense, equidistant from one another. If you embed your neurons in 3D space and actually minimize the physical wiring length, you get predictions like these modules should all live next to each other in the network, and does that match the brain in any way? And what happens?
Certainly, there's been some calculations about white and gray matter that say that if you're doing, because you have like the gray matter is like all the cortical top, which is where the computation happens, and then there's these large cablings that go between them, so the white matter. And it turns out if you try and optimize the wiring length stuff with like some level of interconnection between them, you end up with a ratio of white to gray matter that they claim roughly matches what you find in the brain. So yeah, you can understand those, and then that's already getting you to the point where you're like squashing things into a sheet, and so yes, maybe then you get kind of things that the arrangements you're talking about, but I don't know if anyone's actually done. But you can get, it's super impressive what these Dmitry photography was doing in like early 2000s. Yeah.
Did you draw all those pictures?
Yes.
Make that man a doctor.
Right. I think we're done.
So we'll be up here again.
Oh yeah. We'll be here after the exam ends, which will be maybe from like 4:30ish, something like that. And then we'll be at the pub that you can ask for the address at 6.
Excellent.
Excellent. Sorry, yeah.
I used to.